Okay.
Thank you, Christiana.
Good morning.
Thank you very much. Good morning.
Good morning.
How are you?
Very good, Tommaso. Thank you.
Good.
Welcome to a new episode of Pelo Academy. At PeloBiotech, we can say, yes, we can 3D models. It's kind of a pet project here at one of our founders have worked and introduced several systems working for some new very established companies before. Peter Frost, I will name him because he is here too, because he founded with his partner Lothar Steeb PeloBiotech. My name is Christiane Büchsel and I'm in charge of marketing at PeloBiotech. I'm very happy to be your host today. So welcome to our webinar series about the latest trends of 3D cell culture and models. PeloBiotech is always on the lookout for new 3D cell culture systems. And so what? Guess we found several. One of my favorites, I'm looking now in his eyes, is IVTech, and one of his founders is Tommaso Sbrana. So hi, Tommaso. Good morning.
Hi. Good morning.
Good morning. Yeah, so Tommaso will explain this new technology to you guys and to me as well. Yeah, so he's connected to us via app from Italy. And how's the weather today in Italy? I heard you have sun.
Very good, sunny. Very good and sunny.
Okay, I will have a look at my map. No, in Munich, we do not have sun right now. But 10 minutes ago, we had sun too. Yeah. IVTech have developed a dynamic system, and they made a very big step forward to multi-organ testing and model physiological barriers. So today our topic is advanced in vitro models, a game changer for predictive research. So Tommaso will tell us now how to change the game, right?
I will try.
Yeah, I hope so. So you may share your screen now, please, Tommaso.
Yes, of course. Let me share the screen. Can you see my screen?
Yep. So now we need a full version of it. Yep. Perfect.
Okay, sounds good. So, hello, I'm Tommaso Sbrana. Before to start, I would like to introduce myself. I'm an ex-researcher at the University of Pisa. In the last 13 years, I focused my attention in the development of new tools to overcome the limitation of multiple plate and transfer system. At the beginning, when I was a researcher at the university, I was mainly involved in the technical aspect of development and characterization of this new prototype. Then I shift my attention on the technological transfer, and it has been a natural step at the end of the academic trip found IBTech in collaboration with some of my colleagues. So today I will introduce you IVTech and the products that we offer, and I will also show some applications developed and published by our clients. So let's start. So IVTech has three main goals. The first one is release an enabling technology to develop advanced in vitro model, standardizing the data and increasing the predictivity with human reality. Our history starts in 2014. We were mainly focused on the Italian market, and 2 years later, we released our first starter kit, composed by a pump and one or more chambers, as I will describe to you later. And we signed an agreement with our current Italian distributor, Twinhelix. In 2018, we released our new peristaltic pump called LiveFlow, and we signed the distribution agreement with our current German distributor, PeloBiotech. In 2020, the product's portfolio scenario counted up to 6 chambers and a peristaltic pump, and to increase the distributors network in Europe and also outside the European borders. Our business model is structured on 3 main pillars. The first one is the releasing of the technology through a distributor network. The second one is to offer engineering services in order to help our clients from a mathematical point of view developing scaling models or computational fluid dynamics model. And the third one is to provide biological services in collaboration with biological partners. Our mission is to fill the lack between a 3D and static model and a 5D in vitro model. We want to increase the degree of model complexity by a fourth dimension represented by the dynamicity of the environment. So in our technology, we have a flow of medium which recreates the blood action in human circulation. We are talking about exchange of the nutrient and fresh oxygen, as well as mechanical stimulation of the tissues. This is not enough in order to be predictive of the human reality, because we know that the response to an exogenous stimulus is the result of a cross-modulation between different tissues. So we want to mimic also this scenario in vitro. So we want to add a fifth dimension to our in vitro model in order to study the crosstalk between tissues. Our technology is a very user-friendly technology. It's based on small cell culture chambers. And as you will see, the protocols, the procedures to see the cells inside of our chambers are very similar to the methods that we are used to follow in a multi-well plate or in a transwell system. So let's see, starting from a sterile chamber, we have just to open it. We have to see the cell suspension. In case of a floating scaffold, we can fix it with a plastic ring, and then we have just to close the chamber and clamp it. So now we are ready to place the bioreactor in an incubator environment in order to let the cells attach to the support, to the scaffold, or let the tissue mature in our system. Once the tissue will be ready, we will start the dynamic stimulation. The chambers are standardized in dimension, so we can use a commercial solution for sponge scaffold, hydrogel solution, or even we can use reconstructed tissues. We can also directly print these 3D structures in our chambers or bioprint the tissue using a commercial bioprinter. Once the tissue is mature, we have to join the chamber to a fluidic circuit and a peristaltic pump. We call our peristaltic pump LiveFlow in order to expose the cells to a dynamic stimulation, a dynamic environment. We can have one tissue in dynamic condition, but our chambers are also modular, so we can join different modules as in a Lego strategy approach. Each module is focused on a different tissue, so we have a crosstalk between different organs in order to reproduce a pathway of tissues. This is crucial once we have to test the effect of a drug, because we are going to test the principal effect onto the target organ as well as the side effect onto the related tissue in only one experiment. The chambers are standardized in dimension, so we have Lightbox 1, which reproduces the cell seeding area of a single slot of a multiple plate system, and Lightbox 2, which reproduces the cell seeding area of a transwell. It's important to monitor in real time our dynamic experiments. So on the left, you will see how to sample liquid, avoiding any damages for the tissues and maintaining the experiment online. On the right, you will see how the transparency of our chamber enable the real-time monitoring using an inverted microscope or an advanced optical imaging platform. The chambers are composed by two removable glass discs, one on the bottom side, one on the top side, and in case of a Lightbox 2, a removable membrane. So at the end of the experiment, the user is able to recollect all the biological attached material in order to perform a post-processing in a plate or in a Petri dish. We have three main lines of products on the market. Lightbox 1 is a family of 3 different bioreactors developed for attached cell culture condition. Lightbox 2 has been developed to mimic physiological or pathological barriers. It's equipped with a removable and porous membrane. the porosity and the material of these membranes reproduce the transfer that you can find on the market. And finally, we have also our peristaltic pump lines. It's called the LiveFlow, and we have two different setups, the constant modality and the programmable modality. It's a low weight and compact system which is compatible with the incubator environment. As I told you, Lightbox 1 is a family of 3 different chambers. Each one is compatible with the cell seating area of a different slot of a 6, 12, and 24 weld plate. In case of Lightbox 2, the reference is a transfer. So we have the compatibility with the cell seating area of a transfer of a 6, 12, and 24 weld plate. In the last case, we are talking about commercial standalone inserts. LiveFlow has 2 different setups. We recently released the programmable version. So in this version, the user can decide the time length of the experiment, and then he can also decide 3 or more period in order to characterize this experiment by different flow rates and different direction of the flow. So the pump will adjust these parameters following the setup decided by the user. Once the tissue is mature or the cells are attached to the scaffold, we have to place everything in an incubator environment. So as you can see, the compact dimension of the system and the drawer where we are going to place all the fluidic circuit allows to easily place everything in the incubator. So at this step, at this point, we have just to turn on the pump and switch on the motor. The dynamic experiment starts in this moment. Let's see some application of our technology. First of all, we have to decide which is the suitable chamber and the suitable configuration in order to mimic the functionality of the tissue that we have to study. So in case of Lightbox 1, we have only 1 inlet and 1 outlet. So the liquid will flow from the inlet to the outlet. And in these slides are summarized the main tissues that have been developed and published using this product. In Lightbox 2, we have different configurations. As I told you, Lightbox 2 has been developed to mimic physiological or pathological barriers. We can work with liquid-liquid interface, having the same medium on the apical circuit and in the basal circuit. The membrane divided in 2 sections, the chamber, a apical side and a basal side. But we can work also with 2 different media, one for the top, one for the bottom circuit, and we can work also in air-liquid interface. So we have the opportunity to mimic all the physiological barriers. In the last part of the slide, you will see the perfusion configuration. So we can have a top-to-down perfused configuration. So the liquid will flow from the top side to the bottom side, perfusing a very thick and porous scaffold placed onto the membrane level. This is the suitable configuration for bone application. If we reverse the direction of this flow, we will have the down-to-top configuration. So in this case, we are able to maintain in a floating condition a spheroid which is placed in the bottom chamber. In this case, the membrane will act as a filter in order to avoid the flow of the biological material outside of the chamber. The application scenario is wide. I will briefly introduce the main line of these studies. So let's see permeability studies. So in general, permeability studies performed using a barrier. So we are going to use Lightbox 2. In these slides are summarized the main tissues that have been developed, and I highlighted in red the tissue that has been published. Lightbox 2, as well as Lightbox 1 and the other fluidic stuff, are reusable, autoclavable, and reusable. But we have also disposable material. So in case of Lightbox 2, the disposable material is represented by the membrane. We suggest the use of a transparent membrane in order to monitor in real time the tissue that we are going to develop onto the membrane level, and also the glass disc, which represents the basal surface and the top surface of the bioreactor. In case of a 3D barrier, we can use a scaffold, for instance, an hydrogel solution placed onto the membrane level. In this case, we have to pay attention to the transparency of the scaffold and also the opportunity to recollect the biological material at the end of the experiment. We have different configurations. So the niche configuration is generally used for BBB application. The liquid-liquid interface configuration can be used, for instance, in case of intestinal epithelium model or kidney model. The hair liquid interface configuration is generally used for lung or skin. We can work also with reconstructed tissues. So in this case, generally they are sold in standalone insert, as it is represented in these pictures. And we can directly place this commercial stuff in our Lightbox 2 due to few adapters that we have developed. In case of a hair liquid model, we would be interested also in a direct interaction with the tissue. For instance, in order to perform skin irritation procedure, or to nebulize gas onto a lung model. So we have developed this hair lipid interaction module, which is represented in this picture, and which creates a direct access from the top side to the tissue that is developed onto the membrane level. Once the tissue is mature, we have to join the chamber to a fluidic circuit and a peristaltic pump. In case of a permeability study, we suggest the use of LiveFlow Pro in order to mimic the increase in flow rate related with food ingestion. We will sample the liquid using a three-way valve. Here is an example of a study performed using a Lightbox 2. So in this case, the researchers decided to evaluate the effect of curcumin passage through an intestinal epithelium model developed in a Lightbox 2 compared with a transol. They monitored the cells. In this case, we have a fluorescent staining of nuclei, actin, and microfilaments. And in this part, we have an occludin image. The researchers were able to perform tear measurement and also evaluate the passages of curcumin through the intestinal epithelium layer. The permeability studies can be a first step in a metabolism study. So in this case, we suggest the multi-organ approach. So I'm going to talk about Lightbox 1 and Lightbox 2 because we will join together these chambers in order to have different tissues in connection, in communication. So here I represent the main tissues that have been developed and published using this chamber. And here you can find the main configuration. So in a metabolism study, in general, we use liquid-liquid interface configuration, general to mimic intestinal epithelium or kidney. And in case of Lightbox 1, we can have a 3D scaffold, which can be a porous scaffold or a hydrogel solution, in order to have a 3D model. Again, here we can find the disposable material that we have to consider in case of Lightbox 2 and in case of Lightbox 1. And if the scaffold is going to float in our wet volume, we suggest the use of a plastic disc in order to fix it in position. And this is crucial once we have to perform a real-time monitoring using inverted microscope, because we want to have our 3D model in a fixed position over the time. So again, once the tissue is mature, we are going to join the chamber to a fluidic circuit. And also in this case, the LiveFlow Pro allows to mimic the increase of flow rate in a food ingestion. This can be done for both Lightbox 1 as well as for Lightbox 2. So we are going to establish a setup for each tissue. We would be also interested in increase the pressure inside of our chambers in order to mimic the effect of food ingestion. So we know that the pressure is a key parameter to mimic the real environment in food ingestion. So we have developed LivePA. It's a very last release, and this is a pressure modulator. It increases the pressure inside of a chamber and allows to mimic the variation of environmental condition. Here we have an example of how LivePA interacts with our Lightbox 1, for instance. So as you can see, LivePA is a motorized piston which presses the outlet of a chamber, reducing the lumen. This action will increase the pressure in the chamber. So once we have all the setup, we can join together the different modules in order to have the multi-organ application. As an example of a study in metabolism, I report this paper. In this case, the researchers decided to compare biochemical digestion modality, which is a standard approach, of methylglyoxal, MGO, and they compared the data with a cell-based approach using our technology. So they developed a stomach and intestinal epithelium model, and they used to digest this MGO. Here we can have the data. So in case of biochemical application, a biochemical approach, the main part, the main digestion is performed by the duodenal tract. In case of cell-based digestion, the main digestion is a gastric digestion, and it's also faster. In case of 80 micromolar and 300 micromolar, we can see that after 24 hours, we have a complete digestion of MGO. MGO is a natural compound that we can find in coffee or in bread. Finally, I'm going to introduce you the disease model application. In this part of the study, we have to use a multi-organ approach, and we have very different tissues that have been developed using Lightbox 2 and Lightbox 1. And other different applications are going to be published in the very next period. In this case, we have different disposable materials basing on the different tissues that we are going to take in consideration. So different porosity for the membranes and also for the materials of the membrane. In case of this nylon membrane, we are going to use it to place a fixed scaffold onto the membrane layer in a Lightbox 2, having a perfusion configuration approach. We have to connect the chamber to the fluidic circuit. In this case, we are going to use a LiveFlow to mimic the blood effect, the blood flow in human circulation. And this is crucial also in case of circulating materials such as metastasis or circulating cells such as lymphocytes. And we are going to sample the liquid using a three-way valve. This can be done also for Lightbox 1. And in this case, the LivePA can be used in order to mimic a disease scenario related with an increase of pressure. So we are talking about hypertension in case of vessel or glaucoma, for instance. As an example, I would like to introduce you this paper. So in this case, the researchers decided to use our technology in combination with an IBT chamber in order to mimic to study the obesity. So they mimicked the crosstalk between 3 different tissues, vessel endothelium developed in an IBT chamber, hepatocytes developed in a Lightbox 1 chamber, and human adipocyte fat tissue developed in a Lightbox 2. So varying the percentage of fat tissue, they simulated the normal overweight and obese condition. They evaluated the cell's behavior using, in this case, DAPI fluorescence, and they evaluated also the adipocytes using an Ahoy red solution, and then they sampled the liquid in order to control the biomarkers related with metabolic concentration here, and also pro-inflammatory biomarkers. For instance, they correlated the increase of inflammatory event to the obesity condition. The last study that I would like to talk about is glaucoma. Glaucoma is another disease. In this case, the research group in Genova, Italy, worked for 4 years on this topic. So they have published different papers. So I'm going to introduce you few data, but here you can have all the reference for the papers. So they use our technology in order to develop a model of trabecular meshwork. The trabecular meshwork is a tissue which divided the anterior and backside chamber of the human eye, and it has a very important role in the increase of pressure which characterize the glaucoma condition. So they developed a 3D model of this molecular trabecular meshwork, and they in a physiological condition, and they started to move towards a disease, a pathological condition, using both our LivePA in order to mimic the increase of pressure, and the oxidative stress using H2O2. In the last paper, they used their glaucoma platform to study the effect of iTrub, which is famous for it's a drug famous for its protective effects, and they evaluated the effect of a disease model related with the increase of pressure using our LivePA, and the protective effect of iTrub in combination with our LivePA. So as you can see here, you will see the increase of vitality, the increase of bar if compared to the disease condition. So in conclusion, IVTech products allows to have all the advantages to work with a dynamic model, having the standard cell culture and monitoring protocols. LiveFlow Pro and LivePA are key tools to mimic the variation of the environmental condition related with food ingestion and disease model. And the multi-organ approach is important in order to refine an in vitro model, increasing the predictivity of the human reality. And finally, considering all these characteristics, it's important to have the possibility to real-time monitor and interact with the tissues during the experiment. So for us, it's very important to have, for instance, the hair-liquid interaction model in order to provide an interaction with the tissue in real time. So thank you for your attention. In case of a question, I will be happy to answer.
Thank you, Tommaso. This was a great presentation.
Thank you.
Yeah, I really like your pics and your movies, you know. I always like this. Yeah, we still have some, already have some questions in, And I think we do it this way, that I will collect all the questions. So if you have questions, now type them in. And then I will also introduce you to our CEO, Dr. Peter Frost. He will answer your questions too regarding 3D cell culture. And he has also a lot of experience with IVTech. And so, yeah, one question, Tommaso, would be the first question, the permeability is only for liquid or also for gas permeability? I can't pronounce this word. Permeability.
Well, some of our clients decided to use our chambers in order to study a lung model. So they used to nebulize a gas onto the apical side of this hair liquid interface. And then they studied the permeability of this nebulized drug through the lung model. So the chambers are used in combination with liquid. We can have an air environment, but the exchange, the passages is always a solid which is dispersed in the gas in case of nebulization.
Yeah. Okay. So
Tommaso, did you? Sorry, Christian. Tommaso, did you ever try to use your new pump also to have, I think, a pulse of oxygen so that we can, for example, mimic the breathing, and we have then the medium coming from underneath?
Well, the pump is a peristaltic pump, so it can't be used to move gas. We are developing new application, new items, and we could be able to move gas in a very short period. But at this moment, we are not able to move gas using the products that I show you.
Okay.
In general, you are going to use a pneumatic pump to move gas.
Yeah, that will be good.
Christiane, do you want to continue?
Yes, sure. Then next question would be, if possible, I have another question. Of course you do. How many channels does support your peristaltic pump? Only one or more?
The pump is equipped with 2 removable heads. The heads are autoclavable, so you don't have to set up the tubes onto the head. The head is already equipped with the tubes, and each head drives up to 2 fluidic circuits. So you can work with 4 fluidic circuits. The heads are driven by 2 different motors, so you can have the same velocity for the 4 lines, but you can also work with 2 different velocity, considering a couple of lines.
Okay, so I hope this has answered your question. If not, just type in again and we will refine the answer, of course, Tommaso. And then there's another question. You mentioned that you do not have any experience with lungs yet. Are there any experience yet with the COVID research or with the lung? Is there something in pipeline?
IVTech is an engineering company, so our clients develop the biological application. So in case of lung, our clients decided to use this model to study, for instance, the effect of material in nanofibers nebulized onto the lung. I think they didn't use this model for COVID application yet.
Yeah. Peter, what are kind of experience you have with 3D cell culture models and lung and COVID research yet?
I think I did business development for many companies, and therefore I think there are a few systems in the market, and always you have the question to build up a system which mimics situation in vivo as close as possible. And IVTech offers a very flexible solution, so you can work with primary cells, with cell lines in the way you like to work, and especially if the new system for also the air pulse is available very soon, opens, I think, more application. At the moment, I think most of the application is more in the liquid environment, so that you have, for example, cells on both sides with two different media approach, which already was described very well by Tommaso, has a lot of nice examples. And this is, I think, the main application at the moment, and gives us the possibility to be very flexible, so we can manipulate a lot of different things. And that's also the reason why we don't call the IBT systems microfluidic systems. It's more a mini-fluidic, that also with my, I think, hands, which are not so slim, are able to preparate things. So it's very easy to add cells to seed, also to get parts of the medium out of the running system. That's very convenient to work with that. I have a different question concerning, which came in concerning skin models. So it's possible to use really skin models in your Livebox 1 or 2. Did any of your customers try to work with the Livebox 2 and using endothelial cells in the lower chamber, so that you have a real skin model, more or less?
Well, related with the skin models, we have different applications, different examples. One of our clients decided to use Lightbox 2 in order to mimic the skin, starting from a skin biopsy, both from rat or from human. So this is important. The dimension of the chamber is important in order to allow the use a biopsy, a complex biopsy. In a different approach, different clients decided to use reconstructed tissues from Finion in order to develop the skin model directly inside of the chambers. In order to answer to your question, the cells can be seeded onto the membrane layer or on the bottom chamber, as you prefer. We have different examples of complex barriers where the user is going to see the different cell types on one side of the chamber or also on the other side of the chamber, in order to have communication, a physical communication through the pores of the chambers. So, or the membrane, sorry. The technology is very flexible in order to need the specifications of the clients.
Okay, thanks a lot for this. I think also supports the idea of a very flexible system, depending what your experimental design will be. A different question came in. You offer different membranes. Do you have also a recommendation chart, so that you can tell people who starts using these kind of systems which might be the best membrane to use?
Yes, we do. We have application notes in order to, let's say, to provide a guide for our customers, where we suggest the use of membrane type A or type B. We are not producer of membranes, so we can supply and we can suggest which kind of membrane have to be used. We can also supply to the client. And yeah, we have some specific suggestion basing on the experience that we obtain over the year in case of different issues. Yeah.
Okay, that's great. I think that's also a reason why it makes sense to discuss with us your ideas, because we might have a lot of new ideas which will help you to be successful. On the other hand, I think coming back to Christiana's question, what we are trying to do, we bring also primary cells as well as sometimes special media developments together with the 3D system of IVTech, so that you have the chance not only to get the system, but also we can tell you what type of cells might be useful. We can change the medium composition from a normal FPS containing medium up to defined media, which might have an influence, a huge impact on your experiments. And this we like to discuss with you. Another question came in. Is the live pump, if you plug the live boxes in, also able to change pressure if you use different boxes at the same time, which are connected to each other? I think the change will be then slightly different, but the system has to be the same because it's all in one line. Well, if you use one pump.
Well, the pump is able to adjust the flow rate, not the pressure. So the flow rate is, we offer a product which can work between 50 microliters per minute and 500 microliters per minute. This product allows to work with different chambers connected in series along the 4 different lines. So our clients have an experience up to 4 chambers, so 4 different tissues connected in series. We have tested from an engineering point of view up to 10 chambers connected in series, but of course increasing the chambers, increasing the tissues, it means increase the complexity of the in vitro model. So at the end you have to find a well-balanced a balance between the complexity of the in vitro model and what you're able to control in real time.
Yeah, I think that's completely right. I think it seems to be an easy model, gives us more ideas what happened in our body than a huge complex system where we have a huge black box. Another question came in. You mentioned that also spheres can be used.
Yep.
This scientist has a problem that I want to know. If you have spheres in one of the chambers, how can you prevent the spheres to be disappearing with the medium? This would be also the same case for if you use immune cells, like T cells, together with a cancer model.
Well, in case of spheres, if the sphere is floating, so is not in attached condition, the membrane is a filter to avoid, the membrane in Lightbox 2 in perfusion configuration represents a filter to avoid that the spheres flow outside of the chamber. So in order to maintain the sphere in a normal condition, you have to find the right balance between the flow rate that you're going to use and the shear stress that you are going to develop in using this flow rate. This is the answer can be found using a CFD model, so a preliminary study, a preliminary simulation of the relationship between the flow rate and the mechanical action which is generated with this flow rate, and we can do for the client. So as always, when you start with a new model, you have to perform some trial study in order to evaluate the better condition for your application. So this would be my suggestion. Yeah.
Yeah, that's right. I think that's
in case of lymphocytes. Is a different question, is a different answer, sorry, because you know, the lymphocytes will circulate in the whole circuit. So we have clients in hospital such as in San Raffaele in Milano, which is a very famous research hospital, which they are using circulating lymphocytes in order to mimic leukemia, and they maintain these lymphocytes vital over the time, So they didn't have a problem with the flow rate. They had to find the suitable condition in order to work with human lymphocytes, human-derived lymphocytes.
Okay, I know that working with spheres, so the first question, is not so easy as, of course, membranes and hydrogels can have an influence on the growth of spheres. Did you ever try to use scaffold-based sphere systems? I think there are systems in the market which might fit into a system where I think the sphere has to keep where it is.
One of our clients, if I well understood the question, one of our clients decided to use spheres embedded in an hydrogel solution. So in this case, the spheres were completely embedded in a gel bed. So, but in this case, we are not talking about floating spheres. So in this case, the spheres are attached to the gel. Yeah.
I think there are many different options, and of course, then you have to solve, I think, a few things which will change very fast. Another question came in: for how many lines from the pump can your one live pump regulate the flow rate?
2 lines per flow rate. So you have to think that the head of the pump drives the 2 lines. So each head is driven by a motor. Each motor represents a possibility of different flow rates. So 4 lines is a full system, 2 lines per velocity.
Okay, I hope this answers the question. I think there's at the moment no more questions, but I hope there will more coming in. I think anyhow, if you have further questions, just send by email the question to us. We will then discuss with Tommaso, or Tommaso will answer directly to you. Doesn't matter what it is. And if we have, I think, no idea or no experience, we will tell you as well, so that you know that you have to develop perhaps your own new system. Then if nothing else coming in, I thank you very much for this nice presentation and overview. And I hope that we can start discussion with many scientists, just what you have in mind to do. And I'm quite sure we are able to help you and to bring you some new ideas using new 3D techniques instead of classical 2D.
Yeah. That would be great. And there was only just one question: how many organs can I test in a row? Can I use 6 or liver, pancreas, whatever? Yeah. Well, and probably later lung as well, so that I can also use the multi effect. Or is it just that I need to use 2 and 2 and 2?
No. The answer is some of our clients have 4 organs interconnected in series in only one line. You can increase this number. Of course, you can have also 6 chambers connected in series.
Oh, wow.
Of course, you have to be sure to be able to monitor all these tissues in real time. So increase the complexity of increase the number of chambers, increase also the complexity of the model. So as far as our experience, as far as we are now, the evidence tell us that we can work up to 4 chambers. You can increase this number. It depends from the biological goal that you have.
Mm-hmm. Okay, that sounds perfect. And this really seems to be a very good alternative to animal testing. So I think it's worth a shot.
It can be a help for the animal test, I would say. Not an alternative, but it can be a step forward in order to reduce the number of animal tests.
It's going to be closer to reality. Yeah, that's right.
No, the important thing is that we get an idea what might be the outcome and which kind of compounds we should test with the animals, because then we are able to reduce the number of animals dramatically. At the moment, we have to test many things, which makes perhaps no sense, and we are able to tell people already using 3D techniques.
Yeah, so world is not flat anymore. Going all 3D. So thank you, Tommaso. Thank you, Peter, for this vivid discussion. I really enjoyed it.
Thank you for your kind invitation.
Yeah, sure. And if you have any more questions, we can always schedule an online meeting. If you would like to ask something in private, you would not like to disclose now here in public, I think Tommaso and Peter will be very happy to join the online meeting, and then we can discuss this. Also, if you have any other questions regarding 3D cell culture systems or what kind of cells you should use, just let us know. Yeah, PeloBiotech is one of the most famous, I was already saying, we provide one of the biggest portfolios in the world of primary cells, and we have a very, very good overview of 3D cell culture molds. So just give us a call, write us an email at info@PeloBiotech.com, and also tuned in for our next webinars. There will be also a new website we will see in summer, so just have a look when everything is going again. And I heard we can go on vacation on Italy. Tommaso, is that right?
Yeah, it's right.
Okay, so probably we all come and we'll see the live boxes in real, so that we are not only one step closer to reality, but go to one of your workshops.
Step closer to the ball.
Yeah. We all hope that we are able to have something face-to-face here in Germany as well, because it's somehow missing to have a real conversation with people.
Yeah, that's right. But so far with the online meeting, it's also very nice. So if you would like to see us face to face or just online via Zoom or GoToMeeting or whatever system you prefer, Teams, then just let us know, and we are very happy to get in touch with you. If you have any more questions, just send them directly to info@PeloBiotech.com, and we are here for you. So if you have any more questions, just let us know. Thank you, Tommaso.
Thank you again.
Many greetings.
You for your attention.
Many greetings also. Peter, thank you for everything, and talk to you next month. Okay. Bye.
Okay, bye-bye, Tommaso. Take care.
Bye. Thank you. Bye-bye.
Bye-bye.